they're able to duplicate, double their numbers in 20 minutes. Hardly takes them any time at all to go and make copies of themselves if they're in a good growth medium with lots of food, favorable environment. They just grow in explosive numbers. So part of this efficiency goes into that. Now the operons, we see those in bacteria. We don't see them in eukaryotes like ourselves, but it gives us some understanding of how genes can be regulated. There are insights you can get from this, and so we look at them first. This is one involving what's known as the lac operon, lactose or lac operon. E. coli is able to digest lactose, and so if they're exposed to the milk sugar, they're then able to make enzymes that are suitable for breaking it down. But imagine you've got E. coli in the gut of a vegan person or someone who's lactose intolerant, who has not had lactose for many years perhaps. So the person normally is not consuming lactose, not consuming milk. Would it make sense for his bacteria to go on making the enzymes for breaking down lactose, even if he's never consuming it? No. Bacteria are efficient. They're only going to make these enzymes when they need to. So what they have are the genes for making lactose digesting enzymes all grouped together, and they're switched on when the person takes in lactose. Otherwise, they are inhibited from coming on. So you get this lac or lactose operon. That is including 3 genes for making 3 enzymes that are involved in breaking up lactose. And there is an on switch sitting just upstream of there called a promoter. That's where the RNA polymerase is able to bind and start transcribing all of these genes. But in between the promoter and the genes is another thing, operator. That's a short sequence of DNA, and a repressor protein binds there, and it prevents transcription of those genes unless lactose is present. So normally that repressor is going to have this gene, this set of genes locked down. The operon is switched off by the repressor unless the person consumes lactose, or really the bacteria are exposed to lactose. So you get something like this. We have these 3 genes classified as structural genes, but those 3 are involved in breaking down and digesting the lactose. Lactose itself, is that a monosaccharide or a disaccharide? Disaccharide. And so the first thing you need to do is cut it into the 2 monosaccharides. Now we have in our guts a digestive enzyme for doing that, lactase. Basically that's what this is. Beta-galactosidase will cut the disaccharide into 2 separate units. These help with then further processing of it. But then you've got the promoter upstream of this, and that's where the RNA polymerase will bind and would normally be able to glide right through these and copy a messenger RNA. But if this repressor protein is attached to the operator, it'll get in the way, and this RNA polymerase can't copy the genes. That repressor will be there except if there is lactose in the system. The repressor is produced using a gene that's somewhere else on the bacterial chromosome. This could be thousands of base pairs away, a different gene, a different location on the chromosome. The bacterium produces this repressor protein, and it has an affinity for that operator. It will attach to the operator and block transcription. So that's the situation you have when there is no lactose. If the person has not consumed lactose, the bacteria are not seeing any lactose, then this repressor is active, it attaches, no transcription. And the bacteria is not wasting its energy by making an enzyme it doesn't need. But if lactose does come into the system, it's transformed a bit, and then the lactose binds to the repressor and changes the shape of it so it no longer attaches to the operator. repressor floats around attached to this allolactose. The operator stays clear, RNA polymerase attaches and cruises on through the genes, copying them. So that is a simple way to understand bacterial transcription with genes, and one way it's regulated. There is a more complicated twist on this. It's involving another protein that's able to bind to the promoter and make it faster for the RNA polymerase to attach there. This is not a repressor. This is something that actually helps transcription along. And if that cap is active and promoting transcription, then the genes will get transcribed, copied to make the enzymes. But think about what lactose is made out of. It's a disaccharide. You've got a galactose sugar and a glucose. If the bacterium is getting lactose and it wants to ultimately get glucose to go through cellular respiration, first it has to cut that disaccharide apart. It has a glucose here, but then it has to transform this galactose into glucose. It has to go through some extra steps. And if it has a choice between digesting glucose or lactose, what's it going to prefer? Glucose. So if you provided both things, you provide it with glucose and lactose at the same time, it preferentially uses the glucose. It will go to the lactose once the glucose is used up. But the glucose is an easier thing to digest. It's a quicker energy yield. And so if you have glucose present, then the transcription of these genes for digesting lactose doesn't happen very efficiently. The cell would rather just digest the glucose. So if you have a lot of glucose in the system, then this cap protein is not active, and then transcription is slow and inefficient. You don't get much transcription of these genes in the lac operon, not as long as there's glucose there. So you might have lactose there, so there's no actual inhibition of transcription. But if you have glucose, then the cell is still not going to be really promoting transcription very much, because this cap protein that pushes transcription along is not active. So here's the situation, and I know this is messy, but imagine in the first place we've got lactose. Lactose will knock out the repressor protein, and if there is no glucose, low or absent, then we get an active cap protein, which gives a boost to the RNA polymerase, and it goes screaming down through the genes, copying the genes. But if, let's say, we have lactose there and we still knock out the repressor, but we don't have, but we have glucose, then this cap is inactivated and RNA polymerase doesn't attach very effectively and doesn't really do much transcription. So the cell is modulating its response to as to whether it makes these enzymes, depending on a complex environment. There's a lot of regulation going on here, producing the optimal outcome for the cell. This is going on in bacteria, but you can imagine similar things going on in our cells. So that gives some illustration of the kinds of regulation that do happen in nature. Do you have questions for me on this? Here's one.
The promoter is made of the RNA primer?
The promoter, this is all the DNA sequence. That whole horizontal thing up there with these different colored sections, that's all DNA. So this is all the DNA double helix, but there are different parts that are doing different jobs. So when we look at our DNA, we have these genes, but there's also a lot of regulatory sequences that are nearby, and different proteins are binding to those different sections. So proteins can bind to particular sequences. It's kind of like with the restriction enzymes we were just working with. They attach to very particular sequences of DNA, and they, in their case, they cut the DNA. Well, these proteins are attaching to different parts and doing different things, but like that. So this is some regulation with genes. And in this case, the standard, basically the default arrangement for these genes is for them to be turned off because this repressor is always in the system, floating in the cytoplasm, and it will attach to the operator and suppress these genes, except if lactose is there. There is a different way that this can happen. In this case, we've got this default situation where the inhibition is the default situation unless we have lactose. But there's a different operon known as the trip or tryptophan operon. This is involved in making the amino acid tryptophan. That's one of the 20 amino acids. We always need to have some of it present in the cells for making proteins. And what happens with this is that it automatically, by default, will make tryptophan unless there is tryptophan, and then that suppresses the action of the trip operon. It's basically backwards if you think about the regulation compared to what's happened a lot with the lac operon. So in this case, this one by default is switched on to make tryptophan. But if you think about it, why would the cell make tryptophan, going through all the chemical steps to make it, if it already had tryptophan available? Now, if you just ate a big meal with a lot of protein and you digested that protein and your cells are just flooded with amino acids, including tryptophan, they already have all the tryptophan they want at the moment, and there's no reason to make more of it. But now if maybe all that protein's been used up and they're getting kind of hungry for more protein, then they might start making more tryptophan. So what we have here is something where this trip operon for making tryptophan is switched on when tryptophan is not present. It's switched off when tryptophan is present. It's, again, just a matter of efficiency, thinking about whether the cell really needs to do the extra work to make particular molecules that it already has available. So what we have in this case, there's 5 genes all clustered together. And again, we've got a promoter, an operator, and a regulator. It's similar to what we saw before. And we make this repressor protein that will bind to the operator. It's normally in the inactive state. So normally this would not be attaching, and instead the RNA polymerase can attach to the promoter and run through these genes and make a single long mRNA that then gets cut up to produce the different enzymes from that. Well, if you do have tryptophan available, it's able to bind to that repressor, change the shape of the repressor, so now it binds to the operator, and you're not copying these genes, which means the cell is now not going to be making tryptophan, not until it uses up the tryptophan that's available. And once it uses up the tryptophan, including this part, then the repressor falls back off and it's able to again start copying those genes, making the tryptophan producing enzymes. So again, a different way of regulating genes. Now we don't do this exact arrangement with the operons, but we'll have regulation that is somewhat similar to these. So you think about in our case though, bacteria, they're basically all the same. Maybe slight specialization between different cells, but they're essentially acting as individuals and forming colonies, but certainly they're not differentiating the way we do. You look in a human body, you've got hundreds of different cell types forming all these different organs, and those cells need to do different things. They are producing different chemicals, producing different proteins, and they need to basically be able to make the right ones that they need and not make ones that might be harmful. Your stomach, you get in the inner lining of your stomach, you're producing digestive enzymes. They're in their right place if they're inside the cavity of the stomach. You wouldn't want brain cells switching on those same genes and making digestive enzymes. Your brain would digest itself. Luckily, even though the brain cells possess the same genes, they don't use those genes. All of your cells derive from a single zygote where dad fertilized mom, egg and sperm got together, and that single diploid cell grew up into your whole body with those genes repeated trillions of times over throughout your whole body. But different cells are only using certain genes, and they're not using others. So we switch some genes on and off, and that has a lot to do with how cells get specialized. So when we look at different types of cells, they're different not because they possess different genes, but because they use different ones. They express different genes in a very controlled way, and it's a good thing that they do. So these controls can happen at several levels. You can have control that basically prevents transcription from happening at all, which is probably the most fundamental, most important control. Genes that are never even copied by mRNA to make mRNA in the first place. But beyond that, there are controls that can happen after transcription, but before translation, when the ribosomes get into the act. Then, at the level of the ribosomes, there can be controls then on how active they are with an mRNA. And even after the ribosomes have produced the beginnings of a protein, the protein can be modified, as we've seen like in the Golgi apparatus, and proteins can be broken down. They don't last forever. Messenger RNAs don't last forever. There are different ways where the parts that are being produced by all these steps have limited lifespans and can be broken down. And as a result, if you're going all the way from a gene here to an active protein here, there are lots of ways to dial up or dial down that level of expression, happening at many different levels. We'll look at the transcriptional control here for the rest of this time, and then, like I said, I think we'll be done a few minutes early. This is the most fundamental one. We will look at that, and that's again as far as we get with it today. If you imagine in a eukaryotic cell, we have a nucleus. It contains our chromosomes. You've got 46 chromosomes in there. And then out in the cytoplasm, we have millions of ribosomes floating around. mRNAs are being exported, being read by the ribosomes, producing proteins. Here we've got those 4 different things that I just showed you. Up there we've got the controls on transcription. That's affecting basically whether transcription begins at all, whether the chromatin with the genes in it is readable by the RNA polymerase. And so we've got a variety of ways of controlling whether transcription happens at all. Beyond that, there are further activities that go on in the nucleus after transcription, but before translation happens. That's the post-transcriptional control. Things like, for instance, modifying the RNA with removing the introns and putting exons together in different arrangements. So there is processing going on in there, which is again having an effect on the eventual production of proteins. Then out in the cytoplasm, we're getting control of transcription, controlling how frequently ribosomes are able to attach to the mRNAs and begin translating those. And then even after translation, when we've made a polypeptide, we still have controls over how many of these finished proteins are active in the cell. The proteins may need to be modified. Also, they don't last forever. Not necessarily. They can be broken down. And so the cell is controlling how many of these finished proteins are actually busy in the cytoplasm doing stuff. We're not going to have time to get through all this today, but I want to look at that first one. So here's a way to look at one of our genes. It's similar to what we saw with the bacteria, but they don't normally do the intron exon thing. With bacteria, it's typically just one exon, you might say. In our case, we have these spacers that are built in, separating the parts that are actually going to the ribosomes. So our genes will have a whole mix of exons and introns. You'll have what we call the 5' untranslated region and the 3' untranslated region. These are basically marking off either end of the gene. And in front of the gene, we have a promoter. The promoter includes this region that has a series of T and A bases, generally called the Tata box. So it's a set of maybe like 8 repeats of T and A, for instance. And that is marking this region where RNA polymerase will attach. Then further away, we have regulatory sequences. We have some that are close to the promoter where other proteins will attach that help with transcription. And further away, there are these enhancer regions. These can be a long way away from the promoter. RNA polymerase is going to attach there. And you might wonder how this could have any effect on what happens there. But the fact is, the DNA is bending. It's not like a straight stick. It's able to bend right back, and this part can come in close contact with proteins that are clustered together there. And we'll see that in a video before we're done here. So these enhancers can then control the rate of transcription. So if we're looking at transcription, we'll have what's known as a transcription factor, a protein that will attach in the promoter at that Tata box, and other proteins, other transcription factors, along with the RNA polymerase, will all gather into this spot right in front of the gene. And you'll have activator proteins attached into these enhancers, which can then bend back over and contact this whole region. So we get this complex arrangement set up before transcription begins with these transcription factors and these enhancers. Different enhancers, different proteins, mediator proteins between these can control how frequently the messenger RNA is able to make a transcript. If the cell wants to have millions of copies of a protein, it can use enhancers that really speed up this process and make it happen a lot of times. If it just wants 5 copies of a protein, it'll use enhancers that just make this happen occasionally. So different proteins that are involved in this whole complex can control the number of copies of messenger RNA that come out. Cells are able to dial up or dial down the production of proteins that they need. So I want to show you a video that you've already seen, but before I told you you don't need to know about this first part. Well, now we do, kind of. So let's look at this again.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism.
Okay. So before I kind of skipped over that first part and told you didn't need to really know about those details. Now you kind of do, but just in an overview. I'm not expecting you to identify all these different proteins that we're seeing up there, but understand that when we start transcription in eukaryotic cells, it's a complex process that is highly regulated by these different transcription factors and the enhancers that are able to bind to them. But with all of this, the cell has a lot of options for producing different quantities of messenger RNAs. Beyond that, the DNA itself can be organized in ways with all of those proteins, like the histones. It can be organized in ways that make it easier for messenger RNA to copy or more difficult. It depends partly on how condensed the DNA is. If it is extremely condensed, looped and looped and looped tightly on itself, then that is used basically for long-term storage of the genes and for inactivating them. If it's more loosely wound, where it loops around one of those clusters of histones, but otherwise isn't going into some extremely tight packaging beyond that, then the messenger RNA can attach to it just fine, or the RNA polymerase can attach to it just fine and make a messenger RNA. But what this means is that in any given cell, some of the DNA is loosely organized and some of it is more tightly organized. The cell that is not going to be using certain genes keeps them inactivated by keeping them super tightly wound up. So physically, they're going to be held in a structure that makes them unreadable by the RNA polymerase. And the DNA in certain regions can be chemically altered to make it unreadable. This is something that will be done in certain areas where, again, in a cell that's not going to use a particular gene, the DNA is altered by adding on these methyl groups, CH3s, added on to some of the nucleotides, and genes that have been treated like that are not going to be usable in those cells. So the cell is able to go and make alterations to the DNA. Packaging of the DNA, it can be very loose like we're getting right here. We've got there just a naked DNA molecule. Here it's wound up around histones, the cores of the histone proteins. And in this condition, it's easy for it to be read. But if it gets wound up to this extent, not so. And so the DNA that contains genes that aren't going to be used in the cell will be kept more in this state. And then we get to things like the alternative splicing, where we're actually then modifying the DNA or the RNA. So we produce an mRNA, a free mRNA, and then it is spliced together to produce different versions of a transcript. With that, we then produce different proteins from the same gene. Remember, we have about 20,000 genes, but we're making about 5 times as many different proteins. We do that because one gene provides the information for making multiple proteins. The proteins will be similar. They'll have some of the same parts, but they can be put together, those parts can be put together in different ways, different arrangements. So one gene provides the information to make a whole family of proteins. This is all happening after transcription. Basically, we're going and removing the introns and then putting exons together in different arrangements, leading to different outcomes when we're looking at the proteins. And this was one example I gave you before. The idea that our muscles need to have a particular protein in them, but we have different kinds of muscle, and they use different variants of that protein. We have smooth muscle, we also have skeletal muscle, and they both use a protein that's encoded by the same gene, but it's different versions of the protein. The one that goes into the skeletal muscle is using these exons. Smooth muscle is using many of the same parts, but also some different exons. So you end up getting 2 different but related proteins going into these 2 muscle types. That's an example of the alternate splicing leading to different mRNAs and different proteins. So this is where we then continue with gene regulation, with cells deciding exactly which kind of a protein do they need. And that's as far as we're going to go with it today. I think I'll need to record some more of this in a panopto, and you should be looking at that. Look for further additions to panopto as I try to make up for snow days. We missed the plywood class first, and do all that. With this lab, there's nothing you need to hand in yet. I'll give you directions in class tomorrow, or just what you hand in, and it's not due until next Monday. That'll then count for lab last week and this week. Yeah.
Oh, are we going to be tested on the hallmarks of cancer?
I will, I'm sure, have a question or two on cancer, and so we're going to be looking some more at that. I hope tomorrow, just a second.